Okay, everybody, welcome back. Thanks for coming back and rejoining us here. I'm Jim Way, Executive Director of the American Astronautical Society. Let's get right into our next session. This one is Cell and Microbiology Results. This session includes 6 presentations. There they are listed for you right there. We'll take a quick look at that. Some very interesting looking stuff here. And once again, we're welcoming back our session chair, John Love, who is ISS Research Planning Integration Scientist at NASA. So good to have you back, John. And hey, let's get right into it. Let's start with the session. So Sarah, run that first presentation.
We wanted to understand how spaceflight affects the behavior of cardiac progenitor cell, analyze the proliferation and molecular profile of cardiac progenitor, and characterize the cardiomyocyte obtained. Cryopreserved cardiosphere were cultured by astronauts for 22 days on ISS using the MVP system by TechShot, which can load multiple samples under either 0G or 1G condition. At the end of the mission, we characterize the live cell by examining cell morphology, viability, purity, the expression of cardiac marker, contractility, and calcium transient. And we are also currently analyzing gene expression of a cardiac cell within a short long-term exposure to microgravity. Microgravity affects cardiac sphere morphology and viability. The diameter of the cardiac sphere was measured by ImageJ. Compared to when initially thought, 0G cardiosphere were on average 50 to 100% bigger, while 1G cardiosphere were 50% smaller. Live-handed staining showed that the majority of 1G cardiosphere were fully positive for dead diethedium, while 0G cardiosphere were fully positive for calcine with a minimal amount of dead cell. Cardiomyocyte purity is not affected by microgravity. Cell purity was assessed through cardiac marker NKX 2.5 by RScan. In all conditions, more than 90% of the cell expressed this marker. Cardiac markers like alpha-actinin, cardiotroponin T, and pancreatin were expressed in both conditions, but 0G had more defined and pronounced cardiac structure. Once back at Emory, cardiosphere were transferred in low adhesion dishes and let recover for 48 hours. Video were recorded to compare beating activity between conditions with software MATLAB. 0G cardiac sphere recovered beating activity within 24 hours and had normal traces. Very few spheres in 1G condition recovered beating activity, and the traces were mostly abnormal. Microgravity improved calcium signaling. We analyzed a single cell and we observed that 0G has reduced abnormal calcium signaling compared to the 1G. 0G has also an increased peak amplitude and an increased ramp speed compared to the 1G sample. 0G and 1G cardiosphere were sampled for RNA 3 days after towing and at the end of the ISS mission. Day 3 1G aliquot had 10 times less RNA and lower RIN compared to the 0G cardiac sphere, indicating a high level of damage and cell death. It appears that microgravity is an important factor for cell survival in space. Gene data analysis on the effect of short-term and long-term exposure to microgravity is still currently going on. Data will show what are the most important gene and pathway for cardiomyocyte proliferation and maturation. I would like to acknowledge our collaborators, the people of my laboratory, Emory University, Georgia Tech, TechShot, and Cases.
Hi, I'm Se-Jin Lee, and on behalf of our team, I'll be describing the results of our paper entitled Targeting Myostatin Activin A Protects Against Skeletal Muscle and Bone Loss During Spaceflight. This is carried out under the project Rodent Research 19. We like to call it Sending Mighty Mice to Space. The results of the study were published on September 8th in the journal Proceedings of the National Academy of Sciences. This is me. I was the PI on the project. Emily Germain Lee was a co-investigator. The two of us, together with Adam Lahar, Mike Michaud, and Yiwei Liu, constituted the necropsy team. Other people involved in the project were Renata Ridzik and Dan Youngstrom, who were involved in the micro-CT analysis, Harshpreet Chandok and Josie George, who were involved in the RNA analysis, Tom Stoklasik, who was involved in breeding the mice, Joe Gauguin, who was our collaborator at SomaLogic, where the proteomic analysis was carried out, Liz Warren at the ISS, who was involved in all of the project management, and then Jessica Meir, Christina Koch, and Drew Morgan, who were the three American astronauts who carried out all the work aboard the ISS. The focus of this project was a secreted protein that we named myostatin. We showed many years ago that myostatin normally acts to limit muscle growth, and the dramatic illustration of was that when we genetically engineered mice to lack myostatin, all of the muscles in the body grew to be about twice the normal size. And these mice came to be known as mighty mice. Because myostatin normally acts to limit muscle growth, our work over many years has been to try to develop strategies to block myostatin activity with the hope that this might be effective in treating a wide range of patients with muscle loss. And much of our work is focused on testing this strategy in mouse models of muscle diseases. Now, one model that we've been interested in testing for the past 20 years is looking at the potential benefits of targeting the signaling pathway in microgravity. The rationale being that microgravity would be a great way to mimic the type of muscle and bone loss that you see as a result of disuse. For example, people who are bedridden, people who are wheelchair bound, or the elderly. We were, of course, also interested in testing whether this might be effective in mitigating muscle and bone loss in astronauts during space travel. Well, last year we finally got the chance to test this by sending mice to the ISS. One arm of the study, we sent mighty mice to the ISS to see if the extra muscle mass would be retained when the mice are subjected to microgravity. And in the other arm of the study, we gave normal mice a biologic capable of blocking myostatin from signaling and thereby inducing muscle growth when given systemically to mice. Now, this biologic, because it's capable of blocking other molecules in addition to myostatin, it's also potent in inducing increases in bone density. So with this biologic, we were able to assess effects of blocking this pathway on both muscle and bone. So in my talk, I'll describe a little bit more about the background for this project and then show some of the results that we obtained from this study. Thank you.
Hi everyone, my name is Madelyn Mettler, and I'm just giving you my quick summary on my talk titled The Influence of Material Type and Coating on the Reduction of Biofilm Growth of ISS Isolates. And this was a project that has been going on for about a year now, and it's been taking place at the Center for Biofilm Engineering at Montana State University. And I've been working in collaboration with a few researchers from the Jet Propulsion Laboratory over at Caltech. So I wanted to start off with the project summary and a quick reminder of what biofilms are for those of you who might not remember. And biofilms are communities of microorganisms attached to surfaces. And biofilms can be very difficult to treat. They can cause biofouling, such as clogging of pipes and biocorrosion. And they are sometimes more virulent than the planktonic counterparts of microorganisms. So biofilms are these slimy communities that are hard to clean, hard to get rid of. They can cause a lot of issues, and they can be a threat to human health. So about the project itself, we were looking into a proprietary antimicrobial coating for its ability to mitigate biofilm growth of ISS isolates on water system materials. And we looked into the biofilm growth on Teflon, stainless steel, Inconel, and titanium. And all four of these are materials that are found in ISS water systems. And so far we have evaluated 2 microorganisms. The first being Pseudomonas aeruginosa, which is a bacteria, and the second being Rhodotorula mucilaginosa, which is a yeast. And both of these organisms were found on the International Space Station. And starting with our results summary, we found that the coating significantly reduced Pseudomonas aeruginosa biofilm and planktonic growth for at least 48 hours. So far, the experiments that we've done were only 24 and 48 hours long. And we found that the coating did a really good job of reducing biofilm growth, so the growth of the organisms attached to the surface of the coupons, as well as the planktonic growth, which are the organisms that are just swimming around within the reactor broth. However, we found that the coating did not reduce Rhodotorula mucilaginosa biofilm growth, and in fact, for the most part, the coating resulted in higher biofilm growth and more dense biofilms than the uncoated surfaces. And one of our theories behind this is that the Rhodotorula mucilaginosa, as it is a yeast, it's a larger cell than a bacteria, and therefore it might not be able to nestle down into the surface of the coupon and nestle down into that antimicrobial coating, and therefore it can't, the coating itself can't penetrate into the cell as well as it would for a bacteria. But we're not entirely sure if that's what we're not entirely sure if that's what's happening, but that is our best guess as of now. And our future work includes investigating a fully coated system and low nutrient availability. So we basically just want to take the system that we have currently and do our best in order to model it more closely to the water system of the ISS. And doing so will allow us to extend our experiments on the order of weeks or even months, and therefore we can get a really good idea of how well this coating would work in a long-term system. And with that, I will be happy to answer any questions that you might have in the Q&A session.
This study is about bacterial resistance to antimicrobials in microgravity and countermeasures to this problem. It is a close collaboration between Stanford University and NASA Ames at Moffett Field, and as you can see, several people are involved whose names appear on this slide. Now, bacterial antibiotic resistance is widely recognized to be a serious threat, and this becomes magnified in space because of their long duration, because human immunity is weakened in microgravity, and bacteria may become more dangerous under these conditions. And astronauts are known to suffer from bacterial diseases, for example, cystitis, which is caused by the bacterium UPEC. Now, our discovery of a novel mechanism of antibiotic resistance can control this problem by new drug development, as will be detailed later. This mechanism involves a small protein called sigma S, and what sigma S does is to combine with RNA polymerase and transcribe genes that make bacteria resistant to diverse stresses, antimicrobials, examples of which are given on a slide. Now, what we discovered was that sigma S mediated resistance is due to protection of the very same molecules that antibiotics damage. And therefore, it was reasonable for us to hypothesize that the loss of sigma S should enhance antibiotic effectiveness. This turned out to be correct. Once again, the details. Now, to determine what sigma S regulated proteins account for GM susceptibility of the mutant, we did a proteomic analysis, and it showed that the results showed that GM lowered antioxidant proteins in the mutant when it is treated with, in the mutant alone, not in the wild type. So the result is that the gentamicin treated mutant cannot effectively quench reactive oxygen species. Now that can be a deadly problem because reactive oxygen species are generated during normal metabolism, and inability to quench them, as I said, can be deadly. And so it follows that if we could find inhibitors of antioxidant proteins, sigma S or others, they can be effective drugs to increase antibiotic effectiveness. So we virtually screened over 1 million compounds from the zinc database and found a couple of them were very effective in that their co-administration with gentamicin greatly enhanced the killing of the wild type. I'm not talking of the mutant, the wild type of. And here is a summary of the space experiment. We showed the hardware in which the experiment was carried out. We call it ECMSET, Autonomous Microfluidic Hardware. This was flown to the, with the experiment in it, flown to the International Space Station, and the center part of the slide shows the launch of it from the ISS, and below that are given the various steps of this experiment. And on the right are the results. And the summary of the results is that sigma S does indeed play a role in making bacteria resistant to gentamicin also in space, and that silencing of this protein will enhance GM effectiveness during a space flight, as well as silencing of reactive oxygen defense proteins will provide additional targets or additional means to control this.
During space exploration, astronauts struggle with many physiological challenges, such as cardiovascular and musculoskeletal problems. However, it has become increasingly apparent that astronauts experience changes within the visual system upon their return to Earth. Astronauts have reported vision changes, including light flashes and blurry vision. Retinal cells, rods, cones, bipolar, horizontal, amacrine, and ganglion cells process incoming information from the eye to the brain, and damage to these cells due to microgravity may occur because of altered eye pressure. Damage to retinal cells can produce vision impairment and complete vision loss. Molecular analysis of mouse eyes following spaceflight aboard Space Shuttle mission suggests changes in gene expression and cell death associated with retinal degeneration. We therefore speculated that retinal cell distribution and morphology is affected by microgravity. Histology and immunohistochemistry were used to compare the retinas of Earth-based ground control mice maintained at Kennedy Space Center to those of mice maintained in microgravity aboard the International Space Station. Mouse eyes from the Rodent Research 6 SpaceX 13 30-day mission were acquired from NASA, and the posterior eye cups, preserved in ethanol, were sectioned using a vibrotome. Retinas were incubated with a rhodopsin or cone primary antibody, followed by an incubation with secondary antisera conjugated to Alexa 555 or 488 fluorophore and counterlabeled with DAPI nuclear stain. Sections were imaged via fluorescence microscopy in AmScope software, and morphological data were analyzed using RStudio. Experimental results showed that rod and cone outer segments, immunolabeled with anti-rhodopsin and Alexa fluor-88 fluorophore and anti-cone opsin and Alexa 555 fluorophore, exhibited significant decreases in length in spaceflight conditions compared to ground conditions. Analysis of the widths of the retinal layers revealed in spaceflight specimens a significant increase in mean width of the outer nuclear layer, which contains the cell bodies of the rods and cones. Mean width of the inner nuclear layer, which contains the cell bodies of the amacrine, horizontal, and bipolar cells, was similar in both ground control and spaceflight specimens. Evaluation of the mean number of labeled cells in the outer nuclear layer, inner nuclear layer, and retinal ganglion cell layer revealed in all cell body layers significant reduction in the spaceflight specimens compared to ground control specimens. These results corroborate those of studies that suggest swelling and the presence of apoptotic molecules in retinas exposed to microgravity. This work is important due to its ability to provide insight into common visual dysfunction experienced on Earth. Further, as space exploration gains precedence, it is important to understand the effects on human physiology that ultimately determines the ability of the human body to adapt to environments beyond Earth.
Amyloid aggregation in microgravity. A wide variety of proteins and peptides can aggregate to form linear fibers that can in turn aggregate to form three-dimensional molecular networks that are critical in major diseases, including Alzheimer's and Parkinson's. The aggregation process is not biological in nature and can be duplicated in vitro. However, due to the colloidal size of the structures involved, it's affected by gravity and by fluid convection. Microgravity provides an environment in which thermal convection can be avoided and aggregates remain suspended without mechanically induced convection, which dominates molecular transport even at the molecular level. We used lysozyme as a model for the various proteins that form amyloid in disease. The self-assembly in biology and the origin of life experiment was developed to examine this process under microgravity in a self-contained and automated payload. It provides 9 test cells in which samples can be mixed in microgravity and incubated with individually controlled temperature profiles. The flight experiment demonstrated that amyloid fiber formation occurs in microgravity in a convection-free environment. There are consistent structural differences in microgravity which may help to elucidate the effects of fluid convection in amyloid formation.
All right. Well, thank you very much to all of our presenters. I'd like to bring up our session chair, John Love, to facilitate Q&A. And everybody online, don't forget to ask your questions in the Q&A tool there, right down at the bottom of your screen. Go ahead and submit. Take it over, John. Thanks.
Great. Thank you very much. Again, good morning, good afternoon, good evening, and welcome to the Cell and Microbiology Results Technical Session of the ISS Research and Development Conference. We had some fantastic presentations. I hope you had a chance to take a look at the longer versions as well. And we have a really wonderful set of panelists today, which I will be introducing right now. We have Dr. Antonio Rampoldi from Emory University School of Medicine, Professor Se-Jin Lee from the University of Connecticut and the Jackson Laboratory, Dr. Madelyn Mettler from the Center for Biofilm Engineering at Montana State University, Professor A.C. Martin from Stanford University, Dr. Carl Corgelss from Florida Gulf Coast University, and Dr. Daniel Woodard from the Integrated and Mission Support Services Group at Kennedy Space Center. So thank you so much for joining today. Before I get started with the questions that have been asked during the session, we had one pre-submitted question that is multiple parts. It is actually for Dr. Mettler, so I will go through each of those sections of the question. Was this work done also under microgravity?
No, the whole experiment so far has been done under normal gravity.
Great. Any idea why one of the coating materials mitigated Pseudomonas aeruginosa biofilm growth?
So our best guess so far is as to why it works against the bacteria instead of the yeast. It's just the difference in cell sizes. Like I said in the summary, the yeast are basically bigger cells than the bacteria, so they can't lay on top of the surface as well, and therefore they're not as easily penetrated by the microbial antimicrobial coating. So that's our best guess so far.
Great, thank you. And the last part of the question is, what is the significance of the future plan to use low nutrient availability?
So basically, we're just trying to mimic the ISS as well as we can. And so during the experiments that we're running, we're trying our hardest to grow these really thick biofilms. And so we're feeding them a lot of nutrients so that they're able to reproduce and grow these really thick, visible biofilms. But in reality, on the ISS, there's not a lot of bioburden, and we're looking specifically at the potable water system. So there's not a whole lot of microbes in there. And so in the future, it would make sense to reduce the microbioburden and also reduce the nutrient availability, because the potable water system is pretty clean. So there's not a lot of microbes and not a lot for them to eat.
Excuse me. Are we
Thank
allowed
you.
to ask questions live questions?
Jim, can the panelists ask live questions? Let's go ahead.
Yes, my question to this lady who just spoke, Mettler, I believe is her name, whether she has any notion as to what genes might be involved in the mitigating biofilm formation as a result of this proprietary compound that she tested. And I will shut my...
So the way that the coating works, as it's been explained to us, is there's basically a long carbon chain with like a nitrogen molecule at the end, and that actually penetrates the cells and mechanically and physically kills the cells. So it's not necessarily inhibiting growth on a genetic level.
Excellent. Anything else, Dr. MATIN?
No, it was a perfectly satisfactory answer. Thank you very much. It was good.
Let's move into the questions that have been submitted through the questions tool. The first one is for Dr. Mettler. Was there any difference in reduced biofilm using different materials? Which was the best one?
So we actually found that while there was higher growth on the Teflon for coated and uncoated coupons, we found that the difference between each of the materials was about the same. So the coating reduced the biofilm growth equally across all 4 materials for Pseudomonas aeruginosa. And it actually decreased in its ability to reduce biofilm growth consistently between the days and the materials as well.
Very good. The next question is for Professor Se-Jin Lee. Did the ACVR2B knockout mice, aka mighty mice, not lose any muscle or bone compared to the ground controls? And it has another part, so if you want to go ahead, Professor Lee.
Sure. The mighty mice were mice that lacked the gene encoding myostatin, so it's the MSTN. And those mice started out with about twice the muscle mass, and they retained most, if not all, of that muscle mass in microgravity. The ACVR2B actually refers to the biologic. It's a decoy receptor, one of the receptors for myostatin. And that also was very effective in microgravity, increasing both muscle and bone.
Thank you very much. Another part of this question, have you used transcriptomics to identify which molecular and cellular pathways were resistant to microgravity's effects on muscle and bone growth?
Yes, we did quite a bit of molecular analysis. So we did proteomic analysis of plasma, and then we did RNA-seq analysis of both skeletal muscle and bone. And all of those approaches revealed lots of signaling pathways that seem to be playing a role. Of course, additional experiments would be important to nail down what role these different pathways play. Some of these pathways were already known to be involved in muscle maintenance as well as bone maintenance. So that was reassuring. And then some of these revealed some additional signaling molecules as well.
Excellent. Thank you, Professor Lee. There's another question for you. Do you and your team have any natural or artificial methods to suppress ACVR2B suppression for individuals experiencing low Earth orbit or LEO?
There's been quite a bit of effort in the biotech and pharmaceutical world to develop myostatin inhibitors for clinical use. Almost all of these are biologics, like monoclonal antibodies or things like the decoy receptor. And a lot of these have been tested in clinical trials, but so far, none of them have demonstrated clear-cut clinical efficacy in the various indications that have been tried. So there's still work to be done to try to nail that down.
Very good. Thank you very much. Let's move to the next question. Because it's for Dr. Corgellas. It's partly missing at the beginning. Are there any retinal changes correlated to metabolic changes, for instance, glucose resistance as well as intravitreal eye pressure and were either measured? Do we have Carl Corgelss with us? I'll move to the next question then, see if he ties in. The next question regards biofilm formation. Is for Mettler. Is there any information whether the mechanism of biofilm formation might be different in space versus the ground?
So I'm not entirely sure on necessarily the mechanism, but there is quite a bit of research comparing biofilms in microgravity compared to normal gravity. And there are quite a few differences, including the duration of the lag phase and exponential phase, as well as the different morphologies. There's some evidence that biofilms are more virulent in microgravity and can be a bigger threat to human health. So there are quite a few differences with biofilm growth in microgravity and normal gravity. And we are hoping to look more into these coatings in microgravity once we get a good idea of how it works on the ground.
Excellent. There's another question for you. Can the findings of your work be translated to the prevention or disruption of biofilm formation in organs? And if so, how? I am thinking of biofilm in the lungs of cystic fibrosis patients or on skin wounds.
So since we are particularly in this experiment looking at a material coating, I don't think that that would be able to translate very well to the human body. But there are a lot of different research methods going on with reducing biofilm growth there. And that is more targeted at like the genomics and the proteomics, kind of like Dr. MATIN suggested at the first question.
Very good. And the last question, I don't know if we touched on that before, information on whether the mechanism for biofilm formation might be different in space versus ground.
Yeah, so it is, and there are lots of ways that biofilms are different in microgravity than normal gravity, but we did want to just start off with a simple system and then look more into microgravity experiments since they are typically a little trickier and a little more expensive to run. We kind of wanted to do a proof of concept to start.
Very good. We have another question for Dr. Corgelss. I don't know if he has been able to join us. Are there any known conditions or diseases that could benefit from the results of your research? Go ahead, Alan, if you'd like to answer this question live or make a comment.
Carl, can you hear us?
Yes, yes.
Okay, go ahead and answer, Carl.
Oh, excellent.
All right. Sorry, I couldn't hear the question. I'm reading it now.
I'll repeat it, and we have another one for you as well.
Okay.
Welcome. I'm glad you were able to join us.
Yeah, my
conditions or diseases that could benefit from the results of your research?
I guess retinal degeneration. That's one disease that could benefit from our research. Glaucoma, just certain diseases of the visual system. With our research, we're trying to get a better understanding of what exactly happens to astronauts during their space flight. We use my mouse as a model organism, so it's not, you know, an 100% comparison, but they're a good model organism to use. So I think that would be fair to say. And to the other question, if I don't know if you want to.
Yeah, we have another question. Are there any retinal changes correlated to metabolic changes, for example, glucose resistance, as well as intravitreal eye pressure, and were either measured?
For our research, they weren't measured. I believe that there's another paper that addresses metabolic changes, but just for our research purposes alone, we really looked at the cells in the visual system, but nothing of metabolic changes.
Okay, excellent. I think that is the last of the questions we have received, so I'll go ahead with additional questions for them. Let's start with Dr. Rampoldi, are you with us today?
Yeah.
Welcome, nice to see you. Thank you so much for joining. Your work is very exciting in many regards. You work with simulated microgravity, with true microgravity. You work with culture medium that is carbon dioxide independent, and I think you have largely proven that it works well in space, and you also work with cardiac spheres, very exciting work. So I was wondering, when you were talking about your ISS results, have you compared simulated, what's called simulated microgravity, but actually, analog or modeled microgravity with the random positioning machine, or maybe a bioreactor with your true microgravity results? And do you have any insights or expectations or differences between the two?
We did some experiment with simulated microgravity, and we saw that cells were able to proliferate. They express much more cardiac marker, and they seems to improve the cardiac differentiation. And until now, what we saw is that the cells that were on 0G on a real microgravity, they appear to have the same effect. So the cardiac sphere are much bigger. There is much more expression of cardiac marker. We are also beating much stronger compared to how we should on the ground. So it seems that the real microgravity and simulated microgravity, they have a similar result, but we are waiting now for the RNA sequencing just to compare the result and see exactly how much compared to ground control is the difference in gene expression. So unfortunately, right now, some of the results are still work in progress, but already we have what we already got. It's very promising.
Can I add something to this?
Yes, please.
Yes. There are documented instances in which gene regulation has been shown to be different under simulated microgravity, obtained by using high aspect to volume ratio halves, compared to microgravity during low orbit flight or on ISS. So they are not quite the same thing.
Yeah. And go ahead.
I know this is why we had for the control on the ISS. We had this cell that were in simulated gravity at 1G. Unfortunately, or depending on what you see, the data. If you remember my presentation, the majority of these cardiac sphere, when they were sent back on Earth, were showing to be dead. So our idea initially was to compare the 0G and the cell in a real microgravity and the cell in this simulated 1G condition in space, and then compare any difference in gene expression to see what was exactly the impact on microgravity, and take out any background effect, like, for example, space radiation. Fortunately, we were able to have enough RNA back from this sample at a short time exposure to microgravity after 3 days, and long-term exposure, so at the end of the mission, and we will be able to see the difference in gene expression in both short-term and long-term.
Very good. Thank you very much, Dr. Rampoldi. Anything else, Dr. Madelyn, you would like to add? If not, we can move to you. I'll ask you a question. Your ECAMSAT experiment was really fascinating. A self-contained microfluidic device that is deployed from the ISS, and it is isolated, and the effect of microgravity is happening right there without any of sometimes the other potential influences that may happen on ISS. one of the things that I thought was interesting about your approach is when you were mentioning that a number of antimicrobials that bacteria can become resistant to diverse antimicrobials by effect of sigma S combining with RNA polymerase. But when you look at that spectrum of molecules, it's a wide range: ethanol, starvation, low pH, high salt, etc. That's one part of my question. What's the common denominator? And the second part is the mechanism that protects biomolecules, also of a wide range of types of molecules, cell membranes, proteins, etc. How do you put that all together in the same mechanisms? If you could comment on that. Thank you.
Well, I mean, I'm glad that you enjoyed the talk, and I can see that you have paid attention to it. The range of questions you are asking would take me an hour to respond. However, I can make it as brief as possible. You are absolutely right that what sigma S does is to control a whole array of genes that make bacteria comprehensively resistant. Now, I'll give you one example. You said how. For example, the chaperones are among the proteins controlled by sigma S, and what they do is to disentangle the denaturation of proteins. There is a very involved process by which they do it, and I'm not going to go into that. And I think the other part, there are a couple of things I'd like to mention here. One is that, all we are saying is that the sigma S control mechanism is in addition to the established mechanisms, not to the exclusion of them, but that you can enhance the effectiveness by silencing sigma S or the antioxidant proteins. And as I mentioned, I think in the larger presentation, we do have a small molecules that promise to do that. And finally, the point I'd make is that, if you take a look at the literature dealing with bacteria in microgravity, you would find enormous contradictions. The growth rate, resistance, this and that. Part of the problem is the experimental system. You see, the microfluidic device, the hardware that we used was really very sophisticated, but it subjected the organisms to its own stress. And so we build stress upon stress. There is the stress of the containment, stress of microgravity, stress of the antibiotics. So to tease them out becomes a problem. So one really important issue is to devise a hardware that imposes as little stress as possible. As I mentioned with sigma S, since it is, it's the guts of the bacteria, already it becomes highly, sensitive in the hardware.
Excellent. Thank you very much. We have a couple minutes left, so we'll ask one question to Dr. Woodard. Are you with us?
Yes, can you hear me okay?
Presentation, potential applications of amyloid aggregation to a number of neurodegenerative diseases. And you found some differences between the ground and space and the type of fibrils that are produced from amyloid aggregation. When you talk about the different phases for amyloid aggregation, can you speculate which of those may or may not have been impacted by the near weightlessness of orbital spaceflight, or do you think all of them were, or maybe one in particular? I know you had talked about oligomer formation, linear aggregation, lateral merging, formation of a network, and then stabilization. Do you have any can you speculate on any thoughts about where the impact may have been that produced the shorter, wider macromolecule complexes of amyloid?
Yes, can you hear me okay?
Yes, we can hear you. Thank you.
Oh, okay, very good. Yes, there seem to be two major areas where microgravity, or where the presence of gravity really has an effect. The first is in the degree of fluid convection in the solution. On the ground, because we're dealing with particles that are in the colloidal range, they are at the borderline between individual soluble protein molecules, which can remain dissolved in a solution, and larger particles that will precipitate. And so the only way to maintain colloids of this size in suspension is with forced convection, usually by agitating the reaction container. Obviously, this creates a convection that isn't present in normal tissue formation. The other so the two factors really that are different in microgravity are, number one, it's really the only system in which you can completely avoid convection. Convective flow is the dominant transport mechanism in most systems that are more than a few hundred nanometers in size. And number two, it allows the colloidal sized particles to remain suspended with diffusion only, and thus we can observe the types of molecular formation that might be more likely in tissue where the scale is so much smaller that the large scale convective forces may not be important. And I'd like to also mention that the lack of gravity provides an excellent model where you can study what the effects of these colloidal structures are going to be. We did publish a paper in PLOS One that indicated that the mechanism of cell death in both Alzheimer's and Parkinson's and other amyloid diseases is probably not chemical at all, but is due to the fact that these molecular networks convert both intracellular and extracellular fluids into a gel, preventing convection and thus impeding normal cellular transport processes. So microgravity would also provide an excellent model for distinguishing between the effects of diffusion and fluid convection.
Excellent. I was misreading the time. We have another 12, 13 minutes left. So let's go on with some additional questions. The next one will be for Professor Se-Jin Lee. And I really enjoyed your presentation, fantastic model, very compelling of myostatin and activin protecting against skeletal muscle and bone loss during spaceflight. Wonderful results. I was just wondering, when you're looking at a countermeasure that may be pharmacological or biological, as potentially in this case, or some small molecule, are you looking at all or none responses, or can you somehow attenuate or do the effects somewhat gradual as opposed to all or none? Is that something you can comment on?
Sure. Yeah, we're definitely not looking for an all or none effect. And one of the really attractive features of this entire regulatory system is that it really is dose dependent in many ways. And in fact, you can tune the amount, for example, of muscle mass in various ways, not only by sort of dialing in or out the amount of myostatin you have, but also by dialing in or out lots of different components of this regulatory system in different combinations and so forth. And so I like to refer to myostatin as a molecular rheostat for muscle mass. And so one of the really, again, attractive features of this is that you have the capability of not only modulating the amount of signaling that you get by hitting one molecule, but you can also simultaneously hit other molecules and do it in some kind of combinatorial way. So there are lots of different approaches to targeting this pathway, which I think is one of the attractive features of going after this pathway.
Fantastic. Thank you very much. Excellent.
May I ask a question?
Yes, go ahead, Dr. Madelyn.
This will be about amyloids. And what we saw is that, you know, the size, the fiber size is different. And I suppose there are other differences as well under microgravity compared to normal gravity or Earth gravity. The question is there any implication, evidence, whatever, that one is less dangerous or more dangerous than the other? In other words, would we have less possibility of getting Alzheimer's in space?
I would not suspect that there would be a major impact on the incidence in the human body. That is hard to know, but by and large, the fluid convection in the brain is driven by factors other than gravity, primarily by the pressure differential between the small the capillaries in the brain and the pressure in the intracellular fluid. The brain doesn't have any lymphatic channels, and so fluids flow between the cells. Fluid that's translated from the capillaries flows between the cells and ultimately into the cerebrospinal fluid space. So I think because convection tends to be forced in the brain rather than thermal in nature, that probably the actual risk to human beings would be the same. To us, I think the more critical issue is if we're going to prevent neurodegenerative disease on the Earth, we pretty much have to do it by inhibiting the formation of biomolecular amyloid networks. These networks that basically turn the brain into jello. And because once these networks form, they become cross-linked and extremely stable and can remain in place for decades. So we have to try to piece together the mechanism. You know, how do protein molecules, how are they transported? How do they move through the fluid space? And how do they self-assemble? And one of the surprising things that we did see in the test cells is that when you're doing this experiment on the ground, invariably the protein molecules mix evenly throughout your experiment, your test tube or other experiment cell. But in space, even over a period of the full length of the CRS-5 mission, the very sharp gradients remained in protein concentration. There would be some parts of the test cell where it was highly concentrated and other parts where there was very little. And this probably is something similar happens in the brain, that even over short distances, there's not complete mixing, and proteins can be concentrated in some areas. The formation of amyloid networks is critically dependent on protein concentration. And so this lack of mixing and the potential for the proteins to be very concentrated in very small spaces and then to begin to aggregate is a factor that was obvious even when we looked at the test cells after they returned, that I don't think we really had considered beforehand. And I did want to mention the atomic force micrograph images and so on are in our presentation and a couple of publications. So please feel free to look at the full slide presentation.
Great.
If I
go ahead.
Yeah, a brief follow-up on that question. I believe that amyloids can act as catalysts to promote change in normal proteins to form further amyloids. And of course, as you said, inside the brain, the situation is different. But outside the brain, did this catalytic aspect, do you think, or you have any evidence, did it change under microgravity or is likely to change?
Yes, that's a very important point, because as proteins assemble into amyloid networks, the conformation of the protein molecule is altered in such a way that it becomes non-biological. The protein molecules are distorted. And this does appear to occur as molecules come into contact with the amyloid fibers. That's an area that we would like to look at in future experiments if we can. But I think that the fact that protein molecules and small aggregates are unlikely to precipitate in microgravity would give us a better opportunity to see exactly how they react. But yes, that's just part of the reason that amyloids are stable. The proteins are, in some sense, distorted in shape. The individual monomers come in contact with the growing fiber. They adhere to the surface, and then gradually over a period of time, their molecular structure changes largely from largely alpha helix to largely beta sheet, and they become stable in that configuration. And the way in which this forms is something we'd like to study further.
Great. Anything else, Dr. Madelyn?
So I'm completely well informed. Thank you very much.
Thank you. We received another question through the tool. It's also for Dr. Woodard. Do changes in the fluid matrix interrupt translation and messenger RNA's transport only to amyloid-prone cells, such as neurons or pancreatic cells? And it has a second part.
Yes, that's absolutely true. Neurons are particularly vulnerable to amyloid formation for two reasons. One is that they're very long-lived. If amyloid forms in a skin cell or an intestinal wall cell or something like this, it's going to be sloughed off and die, and before much amyloid can even form. But neurons last their entire lives, and so there are many years for these structures to occur, and that appears to be the mechanism of cell death. Amyloid at one time was thought to have some direct chemical reactivity, but it appears much more likely to us that in neurons, which generally have long axons and dendrites, these axons and dendrites receive vital proteins and other nutrients from the cell body or cell nucleus that have to be transported, as much as a meter or so for very long nerves that extend into the legs. And this is done by means of transport vesicles that move along the cytoskeleton within the neuron. And the formation of even a small area of amyloid gel along the course of an axon can produce a network of molecular fibers that will block the movement of the transport vesicles and thus cut off the more distant portions of axons and dendrites, from critical nutrients. And this may be the explanation for the progression of cell death that we see in Alzheimer's, where the ends of the long processes of neurons tend to die first and the cell body last. I think this happens outside the cell as well, in the intracellular space, where the smaller molecules that serve as cell nutrients are transported. The formation of the so-called plaques from amyloid beta between the neurons may block convective flow of the cerebral or the intracellular fluid during its fairly long path from the capillaries to the cerebrospinal fluid space. And so there too, the diffusion of material, in this case it's not obviously RNA or proteins, but simple small molecular nutrients. They're also the blockage of fluid convection may be a factor. So inside the cell, it's more a matter of mechanical interference with the movement of transport vesicles. Outside the cell, probably more a blockage of normal intracellular fluid convection.
Great, thank you very much for your answer. And I think this brings us to the end of our session. We're pretty much running out of time. I'd like to thank again the fantastic group of panelists that we had and their excellent presentations and the wonderful discussion that followed. And to all the other participants that joined, thank you very much for making this such a productive session. And I hope you can continue participating in the ISSR&D technical sessions. Again,
and we thank you. And we thank you for your excellent chairmanship.
Thank you so much. We'll be in touch with all of you. Take care. Thank you, everyone.
Excellent. Yes, thank you very, very much, everybody. That was very engaging. It was really cool. That concludes this session. We will now take a break for just about the next 15 minutes. So we'll come back at 18:30 Universal, 14:30 or 2:30 Eastern, when we'll get started with our STEM programs and results session. So thanks very much, and we'll see you guys in about 15 minutes.